Good morning, everyone, and welcome. I'm delighted to have you here today for this presentation. Today's topic is validation of microbiological analytical methods. We'll be exploring both the scientific principles behind these methods and the regulatory expectations that guide us. Over the course of this session, we'll cover the key concepts, best practices, and regulatory considerations essential for effective validation. I look forward to sharing insights with you all. Let's get started. Microbiological testing plays a crucial role in ensuring that our products are safe, of high quality, and compliant with regulatory standards. When we talk about method validation, we're essentially confirming that the tests we use are suitable, accurate, precise, and reproducible. This is vital because reliable test methods give us confidence that our products meet safety and quality expectations every time. It's also important to be aware of the regulatory framework supporting these practices. References like USP 1223, 61, 62, 71, 1111, and ICH Q2, R2 provide the guidelines that help us validate and implement these microbiological tests properly. Overall, this slide underscores that rigorous microbiological testing, backed by validated methods and regulatory standards, is fundamental to maintaining trust in our products and ensuring they meet all safety and quality requirements. Today, I want to focus on the key objectives of method validation. First, it's essential to demonstrate that our method is suitable for its intended purpose. This means showing that it can reliably produce meaningful results in our specific application. Next, we need to establish the reliability and consistency of our results, ensuring that repeated tests yield similar outcomes. Confirming the method's accuracy and reproducibility is also critical, as we want to be confident that our results are both correct and can be consistently replicated across different conditions. Additionally, verifying the method's ability to effectively detect microorganisms is fundamental, especially in microbiological testing, where accuracy can impact safety and compliance. Lastly, we must ensure that our method aligns with global regulatory standards. Meeting these standards not only validates our approach, but also supports compliance and trust in our results. Overall, these objectives help us build a robust, reliable testing process that underpins quality and safety. Let's take a closer look at microbial enumeration tests, which are fundamental in ensuring the microbial quality of our products. These tests help us quantify the number of microorganisms present, giving us a clear picture of contamination levels. Moving on, tests for specified microorganisms are also critical. They allow us to detect and identify particular microbes that might pose a risk. Antimicrobial effectiveness testing, or AET, is another key area. It verifies that preservatives in our products work as intended to inhibit microbial growth. Sterility testing is essential, especially for products that must be free from any microbial contamination before reaching consumers. Then we have endotoxin tests, such as the LAL test, which are vital for detecting bacterial endotoxins, especially in injectable products. Rapid microbiological methods, or RMM, are gaining importance because they offer faster results compared to traditional tests, helping us make quicker decisions. Environmental monitoring methods keep a close eye on the manufacturing environment, ensuring that conditions remain within acceptable microbial limits. Finally, it's important to note that many microbiological methods require validation. This is a critical step to ensure their accuracy, reliability, and reproducibility in our quality control processes. All these methods form a comprehensive framework that helps us maintain product safety and quality throughout the manufacturing lifecycle. Now, let's delve into some of the key validation parameters that ensure our methods are reliable and fit for purpose. First, we have specificity and selectivity. These are crucial because they tell us whether our method can accurately distinguish the analyte from other components in the sample. Next, accuracy and recovery come into play. Accuracy reflects how close our measurements are to the true value, while recovery indicates how well we can extract and measure the analyte from the sample matrix. Moving on, precision is essential and includes repeatability and intermediate precision. Repeatability looks at consistency under the same conditions over a short period, and intermediate precision considers variations across different days, analysts, or equipment. Linearity is particularly important for instrumental microbial methods. It shows whether our method produces results that are directly proportional to the analyte concentration over a specified range. Then we have the limit of detection, or LOD, which is the smallest amount of analyte that we can reliably detect, though not necessarily quantify accurately. Complementing that is the limit of quantitation, or LOQ, which is the lowest level we can measure with acceptable precision and accuracy. Robustness is another key parameter, testing how resilient our method is to small, deliberate changes in conditions. This helps us understand the method's reliability in routine use. System suitability and method suitability are checks performed before analysis to ensure the system is working properly, and the method is suitable for the specific sample. Finally, all these parameters form the core of our key validation parameters. They collectively demonstrate that our method is reliable, reproducible, and suitable for its intended purpose. By thoroughly evaluating these parameters, we build confidence in our analytical results and ensure compliance with regulatory standards, ultimately supporting the integrity of our processes and products. Now, let's take a closer look at what this slide confirms about our product. Essentially, it reassures us that our product does not interfere with the growth of microorganisms or the recovery of inoculated organisms. This is critical because we want to ensure that our testing methods remain accurate and reliable. It also confirms that the detection limits are not compromised, meaning we can still detect microorganisms at low levels effectively. The slide also highlights that testing has been conducted with a variety of microorganisms, including E. coli, S. aureus, Pseudomonas aeruginosa, C. albicans, A. brasiliensis, and B. subtilis where applicable. This broad range ensures that our product performs well across different types of bacteria and fungi, covering both bacteria and yeast. Finally, it mentions that the testing adheres to the method suitability test as required by the USP. This is an important point because it aligns our testing with recognized standards, giving us confidence in the validity and robustness of our results. Overall, these points reinforce that our product is compatible with microbiological testing, maintaining both accuracy and compliance throughout the process. When we look at the acceptance criteria, the first point is that the method must clearly detect the target microorganisms without interference. This means our detection system needs to be precise enough to identify the microorganisms we're interested in, without being thrown off by other substances or organisms present. Next, the product should not inhibit the growth of standard challenge organisms. In other words, our method must allow these organisms to grow normally so we can accurately assess their presence and viability. All the compendial strains we test must show positive detection or recovery, confirming that our method reliably identifies these standard strains. It's also crucial that there are no false positives from non-target organisms. We want to ensure that only the intended microorganisms trigger a positive result, preventing any misleading conclusions. Another key point is the method's ability to differentiate between target organisms and background flora. This selectivity is vital because in real-world samples, many other microbes are present, and our method must distinguish the ones we're targeting. We also need to confirm that there's no interference from the formulation ingredients. Sometimes, components of the product itself can affect detection, so our method must be robust enough to handle that. Finally, the method must show a clear, distinct detection or recovery of specific microorganisms. This clarity helps us confidently interpret the results, ensuring the method's reliability and specificity. All these points, specificity and selectivity, are fundamental because they underpin the overall accuracy and reliability of our microbial detection process, which is essential for quality control and safety assurance. Now, let's talk about the criteria we use to evaluate microbial recovery and assess method suitability through recovery studies. When we're testing our methods, we set specific acceptance criteria to determine if the recovery is acceptable. For bacteria, yeasts, and molds, the acceptable recovery range is between 50% and 200% of the inoculum. What does that mean in practice? Well, we spike a known microbial load into the product, then measure how much we can recover. The goal is to see if our method reliably recovers microbes within that 50 to 200% window, depending on the specific test. By evaluating recovery across bacteria, yeast, and mold, we confirm that our method is reliable at different concentration levels. This ensures our testing is accurate and consistent, giving us confidence that we're truly capturing the microbial presence in our samples. Overall, these recovery studies are a critical part of validating our methods and ensuring that our microbial testing is both accurate and reliable. Let's talk about the acceptance criteria for repeatability and intermediate precision. When we look at repeatability, which involves testing within the same day, same analyst, and same equipment, we expect the variation in counts to be very limited. Specifically, the difference between replicates should be within 0.5 log units or a coefficient of variation, or CV%, of 20 to 30%. This helps us ensure that our method is consistent under controlled, routine conditions. Moving on to intermediate precision, which accounts for variability across different days, analysts, or equipment. This is crucial for confirming the robustness of our method in routine use. Here, the variability should be within a 1 log difference or a CV% of 35. This broader scope reflects real-world conditions and assures us that our method remains reliable across various factors. To clarify, repeatability tests involve the same analyst and equipment on the same day to evaluate immediate variability. Meanwhile, intermediate precision involves different analysts, days, or equipment, and it's essential for demonstrating the method's consistency over time and across different operational conditions. Overall, these criteria help us ensure our method is precise and dependable, giving us confidence in our results regardless of who performs the test or when it's performed. Let's talk about the acceptance criterion for microbial detection. Essentially, we need a method that can reliably detect between 1 and 10 CFU, colony-forming units, depending on the method and recovery process used. The key point here is that the lowest concentration at which we can consistently detect the microorganism should be at least 70% of the time across multiple replicates. This ensures that our method isn't just occasionally picking up the microorganisms, but is reliable enough for practical use. Now, when it comes to rapid methods, they can sometimes detect even just a single CFU per test, which is quite impressive and useful for quick screening. Moving on to the concepts of LOD and LOQ, these are critical in microbiology testing. The LOD, or limit of detection, refers to the smallest number of microorganisms that our test can reliably identify. It's like the lower threshold of detection. On the other hand, the LOQ, or limit of quantification, is the lowest level at which we can actually measure the microbial amount with acceptable accuracy and precision. This is especially important in rapid microbiological methods and assay-based tests, where accurate quantitation is crucial for decision-making. Overall, understanding LOD and LOQ helps us ensure that our methods are both sensitive enough to detect low levels of microbes and precise enough to quantify them accurately when needed. Acceptance criterion is all about ensuring that our method stays reliable even when conditions change slightly. It's important that the method remains acceptable if we see minor variations in parameters. For example, we might experience about a plus or minus 10% change in incubation time, or a similar variation in incubation temperature, like plus or minus 2 degrees Celsius. We also need to consider variations in the media, such as using a different manufacturer or lot, and moderate changes in inoculum levels. When we talk about acceptance, the key point is that recovery results should stay within a range of 50 to 200%. This means our method should still produce consistent results despite these small changes. Importantly, there shouldn't be any significant impact on our ability to detect or count microbes accurately. Specifically, the results should stay within a 0.5 to 1 log variation. We also need to evaluate how deliberate variations, like changing the temperature, incubation times, media types, or inoculum sizes, affect our results. This helps us understand the robustness of our method. It's about ensuring our method remains reliable during normal operational variations, including variability between analysts. In essence, robustness is about building confidence that our method performs reliably under slightly different conditions, which is crucial for maintaining quality and consistency in our microbial testing processes. Now, let's dive into what a complete validation protocol should include. First, we have the objective and scope, which clearly defines what the protocol aims to achieve and the boundaries of the validation process. This sets the foundation and ensures everyone is aligned on the purpose. Next, roles and responsibilities are crucial. They specify who is responsible for each part of the protocol, ensuring accountability and smooth execution. Then, we move to method description. This section details the procedures and techniques we'll use during validation, so there's no ambiguity about how tasks are performed. Validation parameters and acceptance criteria are also vital. They define the specific metrics and thresholds that determine success. Without these, we can't objectively assess whether validation is complete. The experimental design outlines how the experiments are structured to generate reliable data, making sure our approach is systematic and reproducible. Data analysis approach explains how we'll interpret the results, ensuring our conclusions are valid and meaningful. Deviation handling addresses how to manage unexpected issues or deviations during the process, which is key to maintaining integrity. Finally, we have the approval and reporting structure, detailing who approves the protocol and how the findings are documented and communicated. All of these elements collectively form the backbone of a robust validation protocol, aligning with our overall theme of ensuring quality and compliance in our processes. Let's take a closer look at the typical criteria we use to evaluate system suitability and validation acceptance. First, recovery within an acceptable range is crucial because it ensures that our test method accurately detects what it's supposed to. Consistent microbial growth is also essential, as it indicates the reliability of the test over time. Repeatability and reproducibility within limits mean that when the test is performed multiple times, or by different operators, the results stay consistent. We also need to confirm that there's no inhibition of test microorganisms, which could otherwise lead to false negatives or unreliable results. Clear identification of the specified microorganisms is key to ensuring we're accurately detecting the right organisms. Lastly, the system must pass the overall system suitability test, demonstrating that all these criteria are met before we can consider the validation complete. These validation acceptance criteria are vital because they underpin the integrity of our testing process, helping us maintain high standards and confidence in our results. Now, let's take a look at the key documentation we need to have in place. First, we have the raw data sheets, which are the foundational records of all the experiments and observations we've collected during the process. These sheets are crucial because they provide the detailed evidence supporting our results. Next are the growth promotion test records, which verify that our media and conditions are suitable for microbial growth, ensuring our testing environment is valid. Then, we have the media qualification documents. These confirm that our media meet the required standards and are appropriate for the tests we're performing. Moving on, the validation summary report offers a comprehensive overview of the entire validation process, highlighting key findings and conclusions. If there are any deviations from the protocol or issues encountered, we document these in the Deviations and CAPA section, along with corrective and preventive actions to address them. Protocol approvals are also essential. They ensure that all procedures have been reviewed and authorized by the relevant authorities before execution. The final validation report wraps everything up, summarizing the validation activities, results, and any recommendations. Lastly, all these elements form the core documentation requirements that not only demonstrate compliance, but also support the integrity and reproducibility of our validation efforts. Connecting all these pieces is vital to ensuring our process is robust, reliable, and ready for regulatory review. Now, let's talk about some of the common challenges we face in microbial testing. One major issue is product interference, which can make it difficult to accurately assess microbial presence because components of the product itself can inhibit or mask detection. Media quality issues are another concern, as using subpar media can lead to inconsistent or unreliable results. Low recovery rates are also problematic, meaning we might not be detecting all the microbes present, which affects the accuracy of our assessments. Additionally, variability in microbial load can occur from sample to sample, making it harder to establish consistent standards. To address these challenges, we have several solutions. First, optimizing our dilution schemes helps to reduce interference and improve accuracy. Performing media qualification ensures that our media are supporting microbial growth as expected, which is critical for reliable results. Conducting parallel method comparisons allows us to validate our methods and ensure consistency across different testing approaches. Lastly, improving sample preparation and handling techniques helps minimize contamination and variability, leading to more trustworthy outcomes. Overall, understanding these challenges and implementing these solutions is key to maintaining the integrity and reliability of our microbial testing processes. Let's take a closer look at some key regulatory standards and guidelines that underpin microbial method validation. First, USB 1223 focuses on the validation of microbial methods, emphasizing the importance of reliable and consistent testing procedures. It provides a framework to ensure our methods produce accurate and reproducible results. Moving on, USB 61 and 62 set the microbial limits for raw materials, products, and environments, helping us define acceptable microbial contamination levels. USB 71 covers sterility testing, which is critical for confirming that products are free from viable microorganisms before they reach patients. Then there's USB 85, which addresses bacterial endotoxins testing, essential for detecting pyrogens that could cause adverse reactions. We also have ICH-Q2, R2, which guides the validation of analytical procedures beyond microbial tests, ensuring our testing methods are robust and reliable across different types of analyses. Additionally, the FDA guidance on microbiological methods validation offers practical insights into establishing sound validation processes in compliance with regulatory expectations. All these references together form a comprehensive regulatory framework that supports the integrity and safety of our microbiological testing. Understanding and applying these standards is vital to maintaining product quality and ensuring patient safety throughout the development and manufacturing processes. Microbiological method validation is a crucial part of ensuring that our products are safe for consumers and meet all regulatory requirements. When we have a well-validated method, we can confidently detect, count, and identify microorganisms accurately. This accuracy is vital because it directly impacts the quality of our products and the safety of the patients who rely on them. By implementing rigorous validation processes, we support consistent product quality and help prevent potential safety issues. In the bigger picture, method validation underpins the entire quality assurance system, ensuring that everything we do aligns with industry standards and protects public health. So, in conclusion, thorough validation is not just a regulatory checkbox. It's a fundamental step in delivering safe, high-quality products. For further support, please feel free to reach out to us. We're here to help with any questions or additional guidance you might need. Thank you so much for your time and attention throughout this presentation. If anything comes up or you'd like to discuss next steps, don't hesitate to contact us. We appreciate your engagement and look forward to working with you. Thank you again.